Hello, thanks for watching this video on the Dudley Lab for cell-free and plant synthetic biology. My name is Quentin Dudley, and I'm one of the newest faculty members at the University of Wisconsin-Madison Chemical and Biological Engineering Department, and we'll be officially opening our lab in the spring semester, January 2024. So in this video, I'll give a brief overview of what we're trying to do, my background, and what the lab's focus is going to be. Let's get started. So for many years, I have been interested in metabolism, this incredible network of enzymes operating within cells, but specifically being able to harness those enzymes as a metabolic engineer to generate sustainable chemicals. And that means we would start with sugar, or in the case of plants, CO2 and sunlight, and link these enzymes together to make useful products, low value, high volume things like biofuels or commodity chemicals, or progressively more valuable and low volume items like valuable polymers or even therapeutics. And so to do this type of metabolic engineering, people have begun to use what's called synthetic biology. And synthetic biology really is just a mindset that endeavors to apply engineering principles of abstraction, modularity, and standardization to make biology achieve useful purposes. And so this vision of using biology for chemical manufacturing is a core mindset that underpins the work the Dudley Lab is trying to do. So how did I get to that mindset? Well, I started and grew up in Southwest Minnesota and did my undergrad work in biological systems engineering at the University of Nebraska. And that was kind of an opportunity to learn what types of research I didn't necessarily want to do, but also to get excited about the discovery process. So I worked in a mammalian tissue culture doing non-viral gene delivery, but also got really excited about applications related to sustainability during my master's work on the life cycle assessment of beef cattle. It was during my PhD at Northwestern University, just outside of Chicago, Illinois, that I really engaged this vision of metabolic engineering. But as you'll see later in this talk, we were doing an alternate approach where rather than using cells to do these transformations, we were using these what are called cell-free approaches. After gaining some expertise during my PhD, I shifted to do a postdoc that rather than using bacterial systems that I'd done in my PhD was shifting to plants. I'll talk a little bit more in the future about why that was really exciting to me. But after completing that postdoc, I spent some time in industry in the Twin Cities at a plant biotechnology company called Calyx. Did a little bit of work in science funding for a philanthropic-backed science funding agency, and that was just prior to starting up the Dudley Lab here in 2024. So I'll talk about how the Dudley Lab fits by talking about what has come before in metabolic engineering. So the conventional approach for metabolic engineering is to engineer well-understood industrial microbes. Think of E. coli, one of the best understood bacteria in the world, or like a baker's yeast, Saccharomyces cerevisiae. And the traditional approach is to come up with your enzyme pathway that converts a substrate into a product. In this case, we'll say we're starting with a sugar, put those genes into that microbe and do industrial fermentation to produce your product. But I've observed during my training that there's some disadvantages of this conventional approach. And one is that you always have this opposing goal of the engineer and the organism. You, the engineer, wants to make large amounts of one metabolite, where the organism wants to grow and develop. It wants to make lots of different things. You have this tension. And it's also more challenging than we expect, that to do high yield production strains in microbes requires years of tuning and optimization. And there are sometimes limits on the toxicity and complexity of metabolite molecules that you can make. That's part of that opposing goal of the engineer and organism. There's also challenges related to the capital needs for these systems. And ultimately, you need some feedstock to feed these systems, which in this case is often sugar derived from photosynthesis and plants. And so there's been a lot of exciting successes in microorganism-based metabolic engineering. But the work that the Dudley Lab is trying to do is to use two experimental systems that address some of these constraints. And these are cell-free systems as well as plants. And so cell-free systems involve reactivating these enzymes in a test tube or ultimately outside of the cell. So that could be, like I said, in a test tube or immobilized onto a surface in a chemical reaction environment. And when you're linking enzymes with together in these cascades or activating complex metabolism in a test tube, you avoid some of these constraints. For example, you have the organism is no longer living, so you don't have as the same competition in terms of goals. You also have shorter workflows than engineering living bacteria, because if you want to adjust something, you can simply pipette the corresponding change into your experiment rather than having to introduce a new piece of DNA into your cell. And this means cell-free systems broadly are more easily amenable to automated liquid handling and the throughput benefits thereof. And finally, because you don't have to keep the cell alive, you have some reduced toxicity constraints for getting complex metabolism to work. And again, that builds off a lot of the work that I did in my PhD. My postdoc, on the other hand, went to the other side of the spectrum, where you still have disadvantages in plants of opposing goals and long experimental timelines. But plants, I think, are really exciting in their ability to pull CO2 out of the air, and they have benefits that I think also address the state of the art, that plants already are some of nature's best chemists with a rich endogenous metabolism. You can imagine that plants are almost like decentralized bioreactors, easily distributable, low capital, scalable. And again, in this case, we're kind of taking out the middleman. We're making our products directly in a plant system where the carbon is coming directly from CO2. So the Dudley Lab is focused in two ways, both on cell-free systems and plant metabolic engineering, and specifically how cell-free systems can make plant metabolic engineering easier. And that's because I see plant synthetic biology as a new frontier for biological manufacturing, with this potential to accelerate a transition to a distributed, sustainable, and post-fossil carbon society. I've always just feel, as I look at a field like this is tobacco growing. This might be a future direction of chemical engineering with the benefits that I briefly talked about. But there's a long way to go to get plant synthetic biology up to where this current state of the art is for microorganisms or even human mammalian synthetic biology. But there are some exciting applications that I think are unique to plants, and I'm going to list through those here. For example, there's been some exciting work on getting plants just grow bigger using synthetic metabolic routes or pathways that are more efficient in dealing with problems that plants have, for example, with photorespiration and accidentally using oxygen instead of carbon dioxide in its main enzyme that pulls CO2 out of the air, Rubisco. There's other really cool ways of getting around these limitations of Rubisco, where we're making completely new enzymes that are pathways that are able to fix carbon dioxide without needing Rubisco at all. Cool areas in having plants fix their own nitrogen, taking N2 gas out of the air, or using plants for distributed bioremediation of soils. Really interested in applications where you can imagine sentinel plants that detect environmental cues, give a signal either to a drone or to a neighboring plant that then enhance the efficiency of our current agricultural systems. You can imagine how this would be done by signaling a farmer to do something, or the plant itself could have a synthetic circuit that, in response to a pest, for example, it makes an agrochemical right there in the field. And finally, many of our useful medicines used in modern society are originally derived from plants, and there are still thousands of plant metabolites that have unknown biological pathways, and our ability to manipulate those, manufacture them, are a really core, cool area of plant synthetic biology. So this painted a couple examples of why synthetic biology is exciting, but there are definitely challenges for our ability to take a metabolic pathway and produce high levels of chemical in a living plant. And those are listed here. First, that plants are relatively slow to work with. It takes, months of time to introduce a stably expressed gene into a plant, to test whether what a gene looks like when it's turned off or when it's inactivated. There's also a limitation in the toolbox of parts that we have for turning genes on and off. I really like this graphic showing a complex wiring diagram of what plant roots might be doing functionally, but we just don't have all of the operators of and or gates to make genes turn on and off in this complex way, in a way that we do have for bacteria. And finally, as I alluded to with the excitement about plants and their natural products, there's still limited knowledge of how many plant metabolites are made, or once they are made, how that manufacturing is regulated. This graphic is showing the decreasing cost in gene sequencing. Has really plummeted over the last year. So we have tons of sequencing data, but we don't know what a lot of that DNA sequence in various plants is actually doing, and we need improved ways to assess what that is so we can make useful molecules. So that's where the Dudley Lab is sitting, at the intersection of a number of goals. First, related to a practical, hard-nosed desire to make molecules. And that's why we're in a chemical engineering department, and that's this focus on sustainable biomanufacturing. We also want to touch and use these synthetic biology approaches for more science and understanding areas, for example, for metabolic pathway discovery, as well as developing tools to control plant metabolism and then ultimately understand how plants natively do these things, what they do. And you can imagine there's many overlapping or ways that these various three goals intersect. So that's broadly what the lab is trying to do. And to get into a few details on the practical techniques that we're using for these three aims, I'll walk through those briefly. So first, we're going to do what's called plant transient expression. I mentioned that it takes months to stably express a plant gene and see what it does when turned on or inactivated. But by using a wild relative of tobacco, this workflow can be to see what a plant does when turned on or off, can be shortened to just weeks. In some cases days, if you're clever about it. So we'll be using this wild tobacco as a experimental chassis. We also have resources for CRISPR-Cas9 based genome engineering, and to be able to do those types of experiments in this tobacco plant without needing to do plant tissue culture, which can, as I alluded to before, can take months of time. So that's really going to be helping us develop some of the plant questions that I talked about in the previous slide. Importantly, we're going to be using tools from cell-free protein synthesis. And this is this phenomena where you can break open a cell that contains the cytoplasmic guts, if you will. And importantly, that has all the machinery for transcription of DNA to RNA and translation from RNA to protein. And by adding a number of other reagents, 30 to 40 in this case, of the 20 amino acid building blocks for proteins, the NTP building blocks for RNA, an encoding DNA template, as well as a number of molecules for energy regeneration, you can make in that test tube proteins directly. And so this cell-free protein synthesis will be a way that we can quickly express various proteins, specifically enzymes, and help to understand what those enzymes, particularly if the DNA encoding them is derived from a plant, and feedback to better understand what targets for transient expression and gene engineering we need to do. Because cell-free protein synthesis is amenable to liquid handling automation, we're optimistic about how we can achieve a higher throughput in characterizing plant metabolism using robots that do the pipetting for us. And in terms of understanding what molecules are made and how they fit into a broader context, we're going to be building resources for using high-resolution mass spectrometry to do what's called metabolomics, understanding what metabolites are accumulating in a given tissue. And because, especially the bottom two, like we're going to be generating large amounts of data, there's going to be opportunities in the lab to integrate and collaborate on artificial intelligence types of applications, specifically for predicting metabolic pathways, for doing protein engineering to make enzymes better, or for optimizing how multiple enzymes behave in a cascade. So that's kind of the overall vision of the lab. And the next slide walks through a few of the available projects that we're imagining spinning up in the first couple years of operation. The first project area is specifically for the plant work that I've talked about. And that's really using these high-ish throughput genome engineering tools to make chassis plants that are more optimized for reconstituting metabolic pathways, both for basic science and for industrial production. And also project areas revolving around better DNA parts for consistent gene expression, as well as installing feedback control into plant metabolic circuits. A second project area for the lab is going to be these cell-free protein synthesis systems that I talked about. Many of the best yielding ones were not necessarily optimized for plant proteins. So we want to make better cell-free systems that are able to express a higher fraction of plant proteins. And we can imagine using that to help Project Area 1, but also specifically for discovery of, in this case, there's some interesting molecules that are nutraceuticals that are of a class of metabolites called flavonoids that could be an interesting way to find useful molecules and showcase how cell-free systems can accelerate that discovery. And finally is to look forward towards not needing a living organism at all and reconstituting enzymes in industrial settings with immobilization to make our molecules directly. And so we're imagining how do you orient enzymes in space and have the toolbox to do that, specifically related to how various energy and reducing cofactors are utilized in these systems. So with that, I will finish up here and say, please feel free to reach out if you have any questions. Looking forward to further conversations. Bye-bye.